All right. Okay. The guy lived from 1854 to 1915, and we gave him credit for chemotherapy because he came up with the magic bullet for syphilis. Who is it?
Ehrlich.
Ehrlich, good. Ehrlich's magic bullet for syphilis. The idea was that it would harm the pathogens, but have little or no effect on the patient. However, his drug was arsenic and sulfur together, so it still might have had some effect on the patient. Then Fleming came along, and what did Fleming discover?
Penicillin.
Right. So here's his bacteria growing on a Petri plate, and notice that there's Staphylococcus aureus, and he's growing them on blood agar because Staph aureus is a pathogen and likes blood agar. However, the plate accidentally got contaminated, and now that you've worked in lab, you know how easily that happens. In fact, even the new plates that I put out for you today, I threw out about 5 of them because they were contaminated with mold. So when you open the plate and pour the agar and you let it settle, you usually leave your lid off just a little bit of a crack because the air needs to come out when it's cooling down. And it's enough for when the air conditioner kicks on, blow a mold spore into your plate. So it's very easy for them to get contaminated. And he noticed a zone of inhibition. Notice the bacteria are not growing around the penicillium mold. That's called a zone of inhibition. And that's what you're going to measure around your discs on your Kirby-Bauer plates. So you did a Kirby-Bauer disc diffusion method. And you're going to measure the zone of inhibition around the discs. Penicillin was not mass-produced, so it wasn't really available until the 1940s. So in the meantime, they used sulfanilamide drug, which was discovered by Domag. So you might see his name on your test, this guy. Sulfa drugs they consider anti-metabolites because it interferes with the metabolic process that the bacteria have to do. They can't make their folic acid from PABA if sulfanilamide is there. Because the sulfanilamide is going to competitively inhibit the enzyme. Penicillin can also work as a competitive inhibitor. Might see Wakesman's name on the test. He gets credit for making the antibiotics. But antibiotics are actually natural agents produced naturally by microorganisms. That's what Fleming discovered. Penicillin is a natural organism producing a natural substance that is anti another living thing. So antibiotic means anti-life. So it's a substance produced by one living thing that's anti another living thing. That's the traditional meaning of antibiotic. Nowadays, we've modified antibiotics. We've made them synthetic or semi-synthetic. We've altered them biochemically. So they're not exactly natural agents anymore. Synthetics, semi-synthetics. Pretty self-explanatory. Selective toxicity is the word you're going to need to know. As well, you're going to need to know the word mode of action. Well, here they're calling them mechanisms of action. Mode of action. Selective toxicity means it's selective about what it's toxic against. If I give Melissa a big old dose of mercury, it's not selectively toxic. It might harm whatever bacterial infection she has, but it's probably also going to harm all your normal flora and all your own cells too. So it doesn't have good selective toxicity. Instead, if I gave her penicillin, penicillin would inhibit the enzyme that makes peptidoglycan. So bacteria are going to be harmed because they have peptidoglycan, but our cells don't. So that is a better magic bullet. Magic bullets are magic because they have selective toxicity. And how do they have selective toxicity? They're going to target something the bacteria has that we don't have or won't be hurt as much in us as it is in the bacteria. So these are your modes of action. When you print out your little sheet to bring to lab tomorrow to do your Kirby-Bauer antibiotic sensitivity test results, you're going to see at the top I have several different antibiotics listed. Those are the ones in the paper discs that we dispensed onto our plates. And it's going to tell you the mode of action for each of those antibiotics. But I love this figure because it's all right there. Now, first of all, let's get this out of our head. This is for viruses over here. So that's not bacteria, that's viruses. So you can almost ignore that for the time being. But remember it, because we're going to do viruses the next chapter, okay? Here's your bacterial cell that we want to target. Can you answer that question, Anita?
Repeat again, what's the mode of action again?
The mode of action is how, write this down because it's the exact wording on your test. The author says mode of action or mechanism of action is how the antimicrobial drug or how the drug exerts its effort against the microbe. How the drug exerts its effort against the microbe. Or the bacterium, if you want to think of a bacterium. So, the mode of action of penicillin is cell wall inhibition, or peptidoglycan inhibition, or whatever you want to call it. The mode of action for penicillin is cell wall inhibition. What's the mode of action for tetracycline? Ribosomes, right. Mode of action is protein inhibition of protein synthesis by harming the ribosomes, by targeting the ribosome. So the target is the ribosome. The mode of action is to inhibit protein synthesis. And here it shows the different modes of actions for different types of antibiotics. So here's your bacterial cell with its nucleoid. Got some ribosomes, plasma membrane, cell wall. Same thing I'm always drawing on the board, only it looks better here. Inhibition of cell wall, inhibition of cell wall synthesis. Here's your cell wall synthesis. This is your peptidoglycan being inhibited. Works really well on gram-positive. Notice this is a gram-positive cell, has a lot of peptidoglycan. Penicillin, cephalosporins, vancomycin, bacitracin. Those are the ones I'd remember for inhibiting the cell wall, the peptidoglycan cell wall. Then you have protein synthesis. I would remember tetracyclines. When I take my lab quiz 5 and my exam 3, that piece of paper that's called antibiotics for you to use, that chart, keep that handy because it's got the information of the modes of action at the top. Ribosomes are inhibited with tetracyclines. Remember, there are side effects to that too. Baby's teeth turn gray. The proteins needed for development of muscles, of bones and teeth can be interfered with. Disruption of cytoplasmic membrane targets the plasma membrane. Polymyxins, those ointments that you put on your boo-boos. Polymyxin bisulfate is one of the ointments you put on your boo-boos. Here you go. These are called anti-metabolites because it inhibits a metabolic pathway. Bacteria cannot make folic acid, which they need for their metabolism, if I interfere with the enzyme that converts PABA into folic acid. So it's an anti-metabolite. In this case, sulfonamides, or sulfa drug, or sulfanilamide, anything with any drug with sulfur in it, you can think of as an anti-metabolite. That was our example for an anti-metabolite. Another way to go is to inhibit DNA or RNA synthesis. I cannot build my DNA copies. I cannot make RNA from my DNA in the presence of these drugs. Nucleotide analogs. This will work for viruses as well as bacteria. What's a nucleotide analog? You know what analogous means? It's like, looks like, resembles, almost the same as. So something that's almost the same as a nucleotide. That's called a nucleotide analog. I give it other names. I call it base analog, because it's actually the base on the nucleotide that's gonna have a phony part. You're gonna put in a phony letter on your nucleotide. Some call them nucleoside analogs, because remember the nucleoside is the sugar with the base attached, the side of the nucleotide. So you'll see them called different things. Base analogs, nucleoside analogs, nucleotide analogs, or base analogs, if I said that enough times. Notice, tuberculosis, unless it's multi-drug resistant tuberculosis, which by the way, remember from the movie, is MDR-TB, that's another superbug. Tuberculosis that's multi-drug resistant is resistant to these drugs that have been successful. Isoniazid was used for tuberculosis in combination with rifampin. So if I use these two different modes of action, I'm taking that mycobacterium and I'm going to hit them with a right hook and I'm going to hit them with a left uppercut. I'm going to hit them two different ways, because just hitting them this way wasn't enough to bring them down. So I'm going to use synergistic effects. I'm going to target them one way and then I'm going to use another drug that's going to hit them again. So that's a cocktail, or two drugs are used in synergism. One drug might enhance the effect of the other drug, for example. So maybe you're going to hit the bacteria with a drug that harms its cell wall, and then you're going to hit it with another drug that prevents it from being resistant. So there's different combinations of drugs that they use. This is key in antibiotic therapy today, because antibiotic therapy doesn't work the way it used to, because the bugs have become superbugs. They're stronger and more resistant to the drugs. So now we know we have to be prudent with use of antibiotics so that we don't